My name is Mohamed Nouri. I'm the Orchard Systems Advisor down here in Kern County, and I'll be moderating the second session here, which mainly dealing with walnut diseases. So our first speaker is Dr. Mohamed Nouri, and he is the Orchard Systems Advisor up in San Joaquin County. And today he's going to be talking about investigating Botryosphaeria, Phomopsis diseases of walnut in San Joaquin and Stanislaus counties. Mohamed, it's all yours.
Okay, thank you very much. Can you see my presentation? Can you see my presentation?
Yes.
Perfect. Okay, thank you, Mohamed, for the introduction. Good morning, everyone. Again, my name is Mohamed Nouri, Farm Advisor in San Joaquin County. Great and pleasure for me to be here today and give you some update on the Botryosphaeria Phomopsis disease of walnut in San Joaquin and Stanislaus County. So this is the second year continuation of my project funded by the California Walnut Board. You maybe got the chance to listen to the first part of my presentation sometimes last year, but we will start from there and continue to the new updated results for the 2021 year. Okay, so as I always mentioned, it is always good to ensure that an orchard gets the best possible disease management practices to avoid major yield losses and problems that could affect the orchard longevity. In walnut orchards, we all know that in the last decade or so, walnut growers in California started experiencing yield reduction due in part to the Botryosphaeria and Phomopsis blight and canker disease, which have been observed increasingly in almost all walnut growing regions. There are many factors that may contribute to an increase in this disease occurrence and distribution, which include the aging of many orchards, walnut scales, as Elizabeth mentioned in her previous talk, the heat, which exposes tree to more sunburn damage that serve as entry points to those fungal pathogens to cause more infections, freeze damage, including what we faced in the last 2 or 3 years, and previous research showed that about 50% of frost damaged shoots and spurs could develop pycnidia in less than a year. Also, the late dormant season rainfall we are experiencing sometimes in the last couple of years, which also increase the chance of the disease spread. So all of what we have back 2 years ago when I started my position, and we started in that time this project, I found growers in San Joaquin County making great effort, spraying many fungicide application per season to control this disease, but with inconsistent results. So with that, we conducted a 2-year survey, mainly to assess the health status of those many aged or mature walnut orchards, and also to check the incidence of those Botryosphaeria and Diaporthaceae fungi. So here we collected and analyzed diseased samples from about 50 walnut orchards in San Joaquin County and other couple of orchards in Stanislaus County. Samples included fruit peduncle blights, twig and spur blights, as well as branch cankers. So survey results showed that Diaporthaceae fungi to be the most prevalent fungal pathogens isolated from those diseased walnut samples. Botryosphaeria fungi have been found occasionally. So here the frequent recovery of those Phomopsis raised the question whether this fungus prevails in the walnut disease complex. Here we believe that the cooler weather condition we have in San Joaquin, also in Stanislaus from the Delta breeze, may favor the germination of those Phomopsis spores, as the optimum temperature for those fungi are a little bit lower compared to the Botryosphaeria fungi. Here, before moving forward in our research project, I want to show the difference between an infection caused by Diaporthaceae and or Botryosphaeria. So when I say Diaporthaceae, That means the Phomopsis. So here, during my farm calls, always I found some confusion between those symptoms caused by those fungal pathogens and other type of dead branches caused by other type of abiotic factors. Sometimes you may be able to recover Botryosphaeria and Phomopsis from the same tissue samples. So symptoms caused by infection caused by those fungal pathogens are very similar in the field, and you can say it's indistinguishable. So here, for example, this is an infection caused by Diaporthaceae fungi. The infection started from the fruit, as you can see here, then killing the peduncle, moving to kill the spurs, and then reach to the branch and cause here an active, if you can see, this is an active infection that will lead eventually to a branch canker. This dead part or this dead spurs, if you can see here, become full of or covered with a dense layer of Pecnidia. And by cutting through a branch or infected branch like that, you will see that the pith inside the branch showed this type of dark black discoloration, which is an indication of an infection. And as the pith canker inside the branch continue growing, or the infection continue growing, the dead part of the branch, as you can see here, will be covered with a dense layer of Pecnidia, same as you see it here. So this picture here, I want to show that by cutting that infected branches like that, leaving that dark discoloration on the pith inside the branch, even here by spraying fungicide, you may not get a consistent results. You may not eliminate the entire infection, as here we believe that the pith inside the branch may provide favorable conditions to those spores to continue germination and causing infection. So here we always ask to be sure that you prune into healthy green tissues, so no spores can be left there that will continue causing infections. Here I want to show you how those fruiting structure produced in infected or dead branches of walnut. You can see here underneath the epidermal layer, you will see those black Pecnidia. They produce Pecnidiospores, and those Pecnidiospores require water or humidity to exude from those Pecnidia and spread to infect other healthy adjacent branches. The other type of dead branches, we usually can find those branches in the lower canopy area of the trees. And we believe that the main cause could be freeze and or shade. And here also the same, by cutting those branches like that through the branch, you will see that the pith inside the branch and the other part of the branch looks healthy and clean. I mean, without any discoloration, which is different from the one I showed before. Same things here, sanitation, very important. We always suggest the grower to be sure to prune those dead branches out of the field, to don't give any chance to those spores of the fungal pathogens to land there, develop Pecnidia that could serve as a huge source of inoculum for future infections. So after knowing the prevalence of those fungal pathogens, we continued our investigation to determine spore or inoculum release pattern during the year. Here we investigated for like last 2 years when and under what environmental conditions spores of the fungi are released in walnut orchard using spore-trapping study. This is a very simple and classic technique of quantifying spores on walnut orchard using glass microscope slide covered with Vaseline. Those microscope slides were changed and analyzed in the laboratory by quantifying the number of germinated spores in a Petri plate like that. Those are germinated spores of Diaporthaceae fungi. So for our study that we did conducted for the last 2 years, for the first year, we conducted this spore trap study in 3 orchards, one with drip irrigation, from where we recovered a very low incidence of or number of spores of Diaporthaceae fungi. and other 2 orchards with the sprinkler irrigation systems, from where we recovered the high incidence of Diaportaceae fungi. Unfortunately, this orchard with drip irrigation system, we were not able to use it for the second year study. So here I want to show you the results of our second year spore trap study, which are very similar to what we got for the first year study. Here in this first graph, you will see we'll refer to the y-axis as the average number of spores captured every 2 weeks, and the x-axis here as the fungal pathogen. So here we have Botryosphaeria fungi with the orange column and the Diaportaceae fungi with the green column. The second graph here, this is the monthly precipitation data from the closest semi-station to this orchard number 1. Same things I will show for orchard number 2. Overall, our second year spore trap study, as I mentioned, were consistent, the results, with those of the first year study. Here we confirmed that the spores were mainly captured during or just following rain events. So as the precipitation increased, spore release also increased, as you can see here. Same things here. Same things for orchard number 2, as you can see here. When we got precipitation, we got spore release. The first things you can also notice, and we confirmed also for the second year, that the average number of spores of the Diaportaceae fungi were much more higher than those of the Botryosphaeria fungi, which confirm actually our 2 years survey results that showed Diaportaceae fungi to be the most prevalent fungal pathogen isolated from diseased samples. The other things we noticed for orchard number 2, and also I will show it for orchard number 1 again, that we were able to capture some spores during or just following the first irrigation of the season. Same things as I mentioned for orchard number 1. And here we believe that after this dry period, the first irrigation of the season may create a microclimate that encourage those infections and the sporulation of the fungal pathogen, like stimulate those pycnidia to ooze spores, and then we were able to capture a few of them. Here, it may be good to consider applying a Botryosphaeria Phomopsis spray before that first irrigation of the season, mainly or especially in orchards or blocks with higher disease incidence. So you will see in my next slides and in my, as a first objective in my research project, that I'm trying to reduce the number of sprays for this disease to help growers. So I'm not trying to increase the number of spray here, but as I always mention to my PCA's growers, if you feel or if you know that you have a high disease incidence, you may consider doing this just before the first irrigation of the season, at least every other row for the next couple of years, until probably and hoping to reduce the disease incidence in those mature orchards. The other things that we also noticed or we confirmed also for the second year, that we got spore release or we captured spores after shredding diseased wood between 3 rows for orchard number 1. I showed these slides last year in several times, and here, as you can see, by shredding diseased woods between 3 rows, you can create a lot of dust, thus creating a lot of spore release and eventually increasing the chance of causing infections in those fresh pruning cuts. I got here also a lot of questions about how to control those infections or how to better manage those canker diseases, especially for those bigger orchard blocks where grower cannot collect all the dead woods and shred them outside of the orchards. Here, we are starting this year new management practices by testing the benefits of spraying fungicide right before shredding or after shredding of prunings, with the objective of possibly reducing infection of pruning wounds and thus lowering the Botryosphaeria and Phomopsis canker and blight disease in walnut orchards. First question also here, how to drive the sprayer probably when we got all those branches here. So our idea is to collect those branches as a windrow every other row. Then we will have the chance to drive the sprayer, then shred the wood, then spray again every other row. Hopefully by that we can get a full spray that we will try to assess the incidence of this disease for the next couple of years doing this new management or different management practices. So it's a simple idea and the different management practices that could help a grower reduce the canker disease in walnut orchards for the long term. All right, moving forward, and after we were able to identify 3 new species of Diaporthaceae captured from our spore traps and also isolated from our diseased samples during our survey, we conducted the pathogenicity study to check the pathogenicity of those new Diaporthaceae species. So we conducted a pathogenicity study on branches, 2 types of inoculation here, one with mycelium plug, and here we were able, in this type of technique or inoculation technique, to inoculate the bark and the wood, and we did not reach to the pith inside the branch. For the other type of inoculation with spore suspension, as you can see here, we did inoculate the entire branches, I mean the pith, the wood, and the bark. Here we did use like 3 to 4 years old branches. This inoculation or this inoculated branches were incubated in the field for up to 6 months. For the fruit inoculation, very simple also, with mycelium plug inoculation, and those fruits were incubated in the lab for 3 weeks. So results are after the incubation period, we found that those new species of Diaporthaceae caused like cankers on branches, as you can see here, and blighted fruits. So after analyzing the data for the walnut branch inoculation with the spore suspension, results of our pathogenicity study showed that all those new species produced cankers, as you can see here, that were significantly longer than those of the control, suggesting that all the new species are pathogenic when inoculated on Chandler and Tulare cultivar. The 2 cultivar tested here were equally susceptible to those Diaporthaceae species. For the mycelium plug inoculation, the results were consistent with those of the spore suspension inoculation, but here the main things we noticed is that the average canker length produced following this mycelium plug inoculation is significantly shorter. This is here an average of 40 millimeter compared to the spore suspension inoculation. Here we got an average of 80 millimeter, which confirm our previous observation that the pith inside the branch provide a favorable condition to the spores to germinate more and cause faster infection. And also here it confirmed previous observation or discussion with Dr. Thames Michelides about how challenging and harder like controlling those pruning wounds infections on walnut compared to those other type of branches for other type of tree crops. The other things or the results from the fruit inoculation also were consistent to those of the branch inoculation. And as you can see here, all the Diaporthaceae isolates were pathogenic when inoculated on fruits, producing a significant wider blighted area compared to the control fruits. Both Chandler and Tulare also were equally susceptible here. Sorry, I did not show this slides. This is the fruits infection or inoculation. Okay, so the other moving forward, and the other good news also in this project, we also moved forward and tested the efficacy of different chemical and biological compound in controlling those Botryosphaeria and Phomopsis fungal pathogens. So we did this experiment on fruits in the laboratory. And as you can see here in those graphs, of the few products tested, Topsin, M, and Rime performed best against the three species of Diaporthaceae, and also New Physiococon parvum, which is a Botryosphaeria fungi. The Ventec product, which is a biological control product, Trichoderma product, have been used in a new study in cherry, in almond, and also in grapes, and performed good on those pruning wound studies. In this fruit inoculation here, or on fruits in walnut, the Ventec and the other biocontrol product that also I used, I call it biocontrol product B, did not perform good, and there was no effect of these two biocontrol products in reducing infection in walnut fruits. We did also conduct pruning wound protection study in the field to test the effectiveness of those same products plus other chemical product and check their effectiveness in protecting those walnut branches from Diaporthaceae fungi. And also we did include here New Physiococon mediterraneum, which is which belong to the Botryosphaeria family. So the biocontrol product B that I did use it for the fruits also here did not provide any disease control, but the other products provide satisfactory disease control with Topsin M as the most efficient product compared to the water-treated control. This is here we did use only water as a control. Luna Experience plus Serenade Optimum and Rime also provided a significant disease control compared to the water control. The interesting things that Ventec, which is the trichoderma-based product, also showed lower disease control compared to the other chemical product, but also significant control compared to the water control here, and can be considered promising candidate in an integrated disease management program against walnut canker disease. So those are preliminary data for the Ventec, and we'll repeat it again next year. Hopefully we'll get a good result because it can be a really good solution, especially for those organic walnut growers. So moving forward in our research project also, and based on what we learned from the spore-trapping study and also from Dr. Themis Michailidis' research about that the infections of mold could start at the styler end. So here we tried together to investigate whether an early spray timing program would be effective to reduce the Botryosphaeria, Phomopsis, and mold incidence in walnut orchards. So here we 3 trials were established in San Joaquin County in 3 different orchards or Chandler orchards, from which we recovered high incidence of Diaporthaceae. Each trial consists of 5-acre blocks, and the rest of the orchards received the grower standard fungicide program used for comparison. The idea was also here to emphasize on using triazole fungicide, FRAC3, and previous research from Dr. Michailidis' lab showed that triazole fungicides are the most effective in inhibiting growth of Phomopsis. So here we started with a bloom spray. We did use Luna Experience plus Serenade Optimum. Then 3 weeks, 3 to 4 weeks before whole split, we did use Marivon and Tebuconazole, and early whole split, like 10-15% whole split, we did use Rime. All application rates here were determined based on the recommended label rates. So to collect data during harvest time, 8 sub-blocks were established randomly in each trial, as well as the rest of the orchards that received the grower standard fungicide program. And blighted fruits or blighted walnut fruits were recorded during the harvest time. So here, each sub-block consists of the area between 4 trees. We relied here on two record time, 3 weeks before shaking the trees, and we call it pre-shaking data collection, and just after shaking the trees, and we call it post-shaking data collection. Results were really exciting and showed a significant disease reduction compared to the grower standard fungicide program for trial number 1. This is here, we have the pre-shaking data, as you can see here, and we have like around 57% disease reduction. And for post-shaking data collection, we got like around 53% disease reduction. Same things, the results were consistent in trial number 2 and trial number 3. As you can see here, a significant disease reduction. Here we will continue our research for the next year and the next probably another couple of years. One of the first objectives for next year is to try to reduce the number of spray. We will keep those numbers of spray as a treatment, and then we'll try to probably compare it with only 2 spray, 1 during bloom and 1 during the whole split time, and also spray 1 blocks with only 1 bloom spray, and see if that could be effective in reducing mold, Botryosphaeria and Phomopsis fruit disease in walnut. Here I want to show you those pictures. I took those pictures October 5th of 2021, like 3 weeks before the shaking of the trees, before harvest day. And as you can see here, a lot of green fruits that drop off in the ground. Those fruits, they got mold and they become infected and they become heavy and hard to blow them out from the walnut. So I talked with the grower always, if you have the tools and the means to collect those branches, to sweep those branches and collect them like during that time, 3 weeks before the harvest, that will be ideal. It will help a lot to avoid mold infections from the ground. Last things to show you here. So as I showed you, when we did collect the data for our spray program, we counted the infected fruits for each sub-block, but also we collect those fruits, like samples from those fruits. And in the lab, I split those fruits into 2 categories. One, I call it in-hulled fruits. As you can see in the picture here, you cannot hold them. They are lighter they are lighter. And the other category, I call it hulled fruits, easy to hold the infected hulls from the fruits. So from those in-hulled fruits, I was able to isolate mold like Fusarium, Alternaria, Botryosphaeria, and Phomopsis. But from the hulled fruits, I was not able to isolate any Phomopsis or Botryosphaeria, which makes sense that, or which make us more believe that the infections of those Fusarium, Alternaria, and mainly the Botryosphaeria and Phomopsis, as I'm investigating on that, could start early in the season, and those fruits got infected early in the season. I want to show you those pictures. This is from those samples. We opened like 50, I think, from each test plot, and this is an example. And you can see that around 60% of those fruits looks good, and probably 20% was infected, and we were able to recover also mold from those fruits, from those kernels. As a summary, we got like new species of Diaporthaceae. We tested the pathogenicity of those species, and all of them were pathogenic to walnut fruits and branches. We found strong correlation between spore release and precipitation and shredding of the wood between 3 rows, or of infected wood between 3 rows. The Topsin M performed good for protecting branches, followed by Luna Experience, Serenadopti, and Rime as well. The biological product Trichoderma could be a promising product in the future, and we'll continue testing that. Bloom spray 3 weeks before whole split and early whole split give us a significant disease reduction, and we'll continue investigating on that. At the end, I would like to thank the California Walnut Board for funding, And thanks for your attention. And if you have a question, I will be happy to get.
Thank you, Mohamed. Sorry.
Oh, I was just going to say, let's jump into the polls real
Yes.
fast. I'll go ahead and read them for us.
Perfect.
Okay, so let me launch this real quick. And it is, which of the following can be considered as a contributing factor to the Phomopsis disease spread? Low-angled irrigation systems, accumulated dead wood on the tree, or all of the above? Make sure to check out, because it says low-angled irrigation systems. Not trying to give it away. Okay, I'm going to go ahead and end the poll, and I will let Mohamed tell us. What's the answer, Mo?
Honestly, I was waiting for this answer to see that probably 50-50, putting all the above or accumulated dead wood on the trees. So it is low angle irrigation system. I should mention this from the beginning, that with my grower cooperator, we did found some spore release during the first irrigation of the season, but after that, there were no relationship between further irrigation in the season and spore release. As my grower cooperator are doing great job in lowering the angle of the sprinklers. And this is what we always ask growers to do. But if you shred the wood, and we need to test more the viability of those fungal pathogens, this is other story on those accumulated dead wood in the ground. Probably from there, they will stimulate some fruiting body to oospores. But doing the best to lower the angle irrigation system in orchards will help a lot to reduce the spread of Phomopsis and Botryosphaeria disease in walnut. So it's logic to solve this, but I will go more for accumulated dead wood on the trees.
Okay, so accumulated dead wood on the tree is actually the answer, right?
Yes.
And that's because he threw a little hitch in there. It's low-angled irrigation systems are okay.
Yes.
Those high-angled irrigation systems you need to watch out for.
Perfect. Perfect.
Okay. And we have one more for this topic. The greatest spore release of Diaporthaceae and Botryosphaeriaceae species occurred during rain events, during shredding and infected branches between the tree rows, or all of the above. I'll give them a little more time. Thanks so much for taking part in these polls. Good way to exercise your brain. Okay, I'm going to go ahead and end the poll, respected time, and we'll share the results. Okay, Mo, let us know.
All the above, the answer.
All of the above is correct, yes?
Yes.
All right. Thank you very much for that.